Right, well, good afternoon everybody. Welcome to the Rothamsted seminar series. I'm delighted today that we have Andrew Mead with us to give the seminar. I'm going to give a little bit of background on his career today. So Andrew, as you might have been unsurprised, is a PhD in statistics. This was at the University of Bath in the middle of the 1980s. But he also had a one-year work placement for Unilever research on Merseyside as part of that first degree. He then went on to do an MSc in biometrics. That's the application of statistical and mathematical approaches to the biological sciences. And this was done at the University of Reading, completing that in 1987. And then was in a very good position with immediately after completing his masters. He was able to walk into a scientific officer position at what was then known as the AFRC, that's the predecessor of the BPSRC, Institute of Horticultural Research at Wellesbourne. Now many of you know that Wellesbourne in the subsequent years underwent many reiterations, organisation and everything, and eventually ended up inside the University of Warwick, and Andrew ended up in the School of Life Sciences, and he then spent a number of years there. But he finally decided to leave Warwick and was tempted to Rothamsted to actually head up the Applied Statistics Group, arriving on the 1st of April in 2014. So as he points out to me, that's exactly 4.5 years ago today.
Though I didn't actually manage to arrive on the 1st of April because I smashed my collarbone 2 days before it.
Oh, that's okay. In terms of awards, this is a very interesting award. Andrew was the first recipient in 2006 of the Award for Outstanding Contribution to the Development of the International Biometrics Society. And then after that, he's actually served within a couple of years as both the president and the vice president of that new society. Andrew has been the co-author of 2 statistical textbooks, which are well used by the community, and he's also been the author of over 100 papers, most of which are published in applied science journals. And his research interests have been maintained throughout this in the area of effective application of statistical methods to enhance biological science research. And that really is going to be the topic we're going to be talking about today. So the floor is yours, Andrew.
Thank you. Thanks, Kim. Yeah. Okay, so I'm actually going to start with a bit of history. If it works, yes, okay. Because we're currently in the 100th year of statistics being applied at Rothamsted. Most people will probably know that R.A. Fisher was the first statistician. He was appointed by John Russell in 1919 as it says here, a temporary post to see if he could find anything useful that mathematics could do to get anything interesting out of the long-term experiments. And Fisher's work, he started off with lots of papers, or a number of papers that looked at variability, trying to understand variability in crop yields across a number of experiments. But his early interactions with the LTEs and other experiments here at Rothamsted really led him on to developing the principles for designing experiments that we now use very heavily, and which I'm really going to talk around in the rest of my talk today. So a series of publications, as you can see at the bottom of the slide here, a letter to student W.S. Gossett, who was working for Guinness, his book on statistical methods for research workers that established a lot of these ideas, including analysis of variance, and then a paper in 1926, and then a book in 1935 on the design of experiments, which really brought together a lot of the ideas that we now use today, or we still use today, in terms of thinking about designing experiments. And I meant to say that we will be celebrating the centenary of Fisher's arrival next year. We're hosting a conference next July in this auditorium of the International Biometric Society, the Channel Network conference, which will include a Fisher Memorial Lecture and a celebration of that centenary. So, Fisher's ideas, Fisher's principles for the design of experiments. What were they really about? Well, really, one of the things he stated was that they were about providing reliable estimates of errors in field trials. Of course, they do a lot more than that. But that then provides us with the basis for assessing whether treatment effects are real, for assessing whether what we're interested in, the treatments we're applying, are actually doing anything useful. But it's worthwhile saying that those ideas that Fisher brought together in the early 1920s and into the 1930s weren't particularly new ideas. People knew about the idea of replication, even though if you looked at a broad book, you'd probably wonder whether we really did know about the ideas of replication. But the ideas had been known about before Fisher brought them together. What Fisher did was to develop the idea of analysis of variance as providing a coherent theory that allowed us to bring these concepts all together and provide us with a way of designing good, effective experiments to answer the questions we were interested in. So the principles that Fisher established were the idea of replication, providing us with the improved precision of the information we were getting from our experiments. Initially, the ideas, I guess, related to t-tests, but Fisher developed those ideas to allow us to provide better comparisons within analysis of variance. The idea of randomization effectively was important from a mathematical viewpoint of allowing us to make assumptions about independence in the way we constructed our analyses, the basis for most of the tests of significance that we still apply. Early trials were often systematically arranged, and that systematic arrangement, as well as having potential problems with independence, also, of course, potentially meant that differences between treatments weren't actually due to the treatments that we were applying. So that's the other aspect of randomization. Blocking or local control, another important idea for taking a trial, separating a trial into areas, groups of plots, groups of resources that we expected to behave similarly. And as I'll try and touch on later on in the talk, there's all sorts of different reasons why we might want to think about blocking. I think blocking is still probably one of the least understood principles in a lot of ways of thinking about experiments. People don't always understand why we need to block or what we need to block for and the purpose of including blocking in the design of an experiment. Excuse me. And the other idea, which is often not really thought of as the principle of design, I guess, is the idea of factorial treatment structure. The idea that we can actually answer more than one question at a time in an experiment. That we can do that often with increased power or using fewer resources, so we can make a very efficient use of our experiments to answer lots of questions. And even in the very early development that Fisher put together, he started talking about incomplete factorial structures and all sorts of other complications, again, some of which I will try and introduce during the rest of this talk. So Fisher's principles established in the 1920s, 1930s, but are still important today. To just continue the sort of slight historical introduction, Fisher left Rothamsted in 1933, but the work on design continued throughout the most of the next, whatever we are, 85 years to the current day. If you look at the statistics roll of honour, the board that some of you may have seen in the corridor in the Borden building, a fraction of which I've shown on the right-hand side there, we find a whole range of famous statisticians who have contributed to ideas about design. Yates was Fisher's successor, and this text, TC35, no, we don't have a pointer, okay. TC35 is referred to as TC35 rather than its formal name, but it's a very well-used paper book about the design and analysis of factorial experiments. Cochrane, Kempthorne, and Canui were all relatively short-term visitors to Rothamsted, but I think their time at Rothamsted and interacting with Yates and others led to each of them producing a fairly well-used book on design. Cochrane and Cox is one that a number of us statisticians use regularly. Others, Patterson and Kempton, worked at times here and then went off to the Scottish agricultural system. And you can see various other developments of statistical design ideas here. I guess worthy of note are those the developments by both John Nelder and Roger Payne. John Nelder was the third head of statistics at Rothamsted after Fisher and Yates. And they, I guess, were responsible for a lot of the development of computational tools for design that we still use now and that have actually enabled us to extend the approaches that we can take to the design of experiments from those ideas. I think that Fisher's ideas sort of established the principles of how we should think about designing experiments. But I guess a lot of the early development of the design of experiments was constrained by the fact that we were working without very much computational power. A lot of those early experiments would have been analysed using something like the Millionaire, and there's Frank Yates using it. It's still, I think, the Millionaire is currently in the museum in the old sample house. Designs therefore had to be simple because we couldn't do complicated calculations. We were coping with relatively simple computational tools. As design ideas developed through the 1940s and 1950s and early 1960s, they were taken over by the mathematicians who came up with clever bits of mathematics, but which were actually pretty useless in terms of applying to real problems. So clever maths that gave us developments in design, but not really about the practical design. I think the development then of the software, such as GenStat here, developed by when Nelder moved to Rothamsted from Wellesbourne in the early 1960s, started to allow us to think about more complex design ideas. So Nelder, working with Graham Wilkinson and Roger Payne, developed a general ANOVA algorithm, which is what we still use in GenStat to this day, separating block and treatment structures, an idea that hopefully you'll see as I develop my ideas a little bit further later on in the seminar. And then more recent developments have included work, including by Robin Thompson and Sue Wellham here at Wellesbourne, on the Vemel algorithm that allows us to analyse linear mixed models and allows even more flexibility to the way we think about designs. So what I'm going to try and illustrate in the rest of this talk is how the modern biological methods, modern methods of research potentially introduce more constraints, potentially introduce more challenges to the way we actually might look at designs, but that Fisher's design principles are still really very relevant. We still use them to this day to construct designs, but the ideas and the tools that we have developed based on those now allow us to be somewhat more imaginative in the way we construct designs, that we're not constrained by having to have simple designs that can be analysed using simple computational tools, but we can now think of quite complicated ways of tackling the real problems that we're interested in running experiments. So what I want to talk about is really six design ideas that I think are design ideas that we can use to help us enhance the way we design experiments, to enhance what we can get from our research programs. So that's the idea of the design process. And the design process is what I've tried to illustrate in this diagram here. So that's the idea that if we're designing any experiment we want to design, we ought to start by thinking about the aims and objectives. Having thought about those aims and objectives, we then really split the problem into two parts. At least this is the way we should think about it. That's the idea of thinking about the treatments, the treatment factors that we need to answer the questions that we're interested in. And from those, identify the treatment combinations that are necessary to be included in an experiment to answer those questions. And separately, to think about the resources that we have available to do the experiment. And identify potentially the constraints that we might have on those resources that that influence the way we're able to use those resources. And my arrows on this diagram are really trying to illustrate that whilst we're thinking about those two separate elements, the decisions that we might make about the resources, what we identify about the resources, may influence how we have to think about the treatments and the treatment structure. But equally, that the decisions we make about the treatments and the treatment structure may influence the way we can use the resources that are available to us. And again, hopefully I can illustrate that in some of the examples I'm going to talk through in the rest of this afternoon. Well, the next few minutes, not the rest of this afternoon. I'm not keeping you that long. It's statistics, and I know you'll get bored if I keep you too long. So, and I should say, you know, as it sort of says at the bottom of the slide there, once we've thought about the treatments and thought about the resources, the interesting bit, at least from a statistician's viewpoint, is putting those together to construct an interesting design that actually allows us to answer the question. But we need to go through these steps, and those steps need to be thought about in collaboration with you, the scientists who are interested in answering the questions, to come up with the best design for your experiment. So that's the idea of the design process. Factorial structure I've already mentioned. I know that already I have this reputation that replication is a strange word that I have all sorts of interesting ideas about. Like whether we need any. Well, we need one, as Ian and I were talking about the other day. We certainly always need one replicate, or maybe we don't. We'll see in a minute. Ideas of constrained randomization, which is really an idea that is starting to become, I think, useful very widely, but comes predominantly from medical research. Again, I'll illustrate that as an idea shortly. Multi-phase and multi-strata issues are things that we need to think, ideas we need to be aware of, and potentially very powerful ideas for modern biology. Again, I'll try and illustrate that. And I'm going to touch just briefly on some analysis issues towards the end. So what I'm going to do for the next 20 minutes or so is to really talk through a series of recent examples which I hope will illustrate how I've applied these kind of thinking, these thought processes, these ideas, across a range of different environments to solve real problems and come up with experiments that are effective and efficient at using the resources that we have available. What I'm not going to touch on particularly this afternoon is the fact that designing any experiment, we should, or we hopefully will, have information about the variability and about the biological effects that we're interested in assessing. And that's an important part of designing any experiment and relates to the amount of resource that we need, or the amount of replication, the way we might set up our experiment. But I'm not going to go into the details of that this afternoon. So
First, a challenge. This is a glasshouse screening assay. It was an experiment an idea for an experiment that Stefan Hefferly and Kathy Thomas brought to me, I guess, about 6 months ago. Part of the AFSIS project looking at quantifying the relationship between wet chemistry measurements and spectral data. So it's a plant assay to assess variation caused by soils, so looking at 30 different soils from the collection that they're making. And they came to me and said, well, we think we've got 7 different nutrient treatments that we're interested in trying to assess within this study. So a 0 nutrients and a full standard nutrient set, and then a series of nutrient treatments with 1 element omitted from the set. So that was the starting point. They had an experiment they wanted to design. How could we design it effectively using the resource that they had available? Initial discussion about the treatments identified that really the full and the standard, the 0 and the full were the primary treatments they were interested in, with less interest, if you like, in the omission treatments, the minus treatments of nutrients. Again, the standard issue often with designing an experiment, we've only got a limited amount of resources, so they only had soil to have 20 pots, 20 replicate pots for each of their soils. So it gave us a total size of the experiment.
And in this case, we did have some information from a previous trial that told us something about the amount of replication we might need to be able to answer, to look for sizes of differences that we were interested in. We were also limited by the space that we had available to design this experiment. So we had 3 glasshouse compartments. Each of those glasshouse compartments had 4 benches. Each of those 4 benches could have up to 5 trays, and you could have up to 12 pots per tray. So lots of constraints that you need to try and think about as to how you put this set of treatments, 30 soils in combination with 7 nutrient treatments, how you might put that into a glasshouse to answer the questions about the differences between the soils and the differences between the nutrient treatments. So how do we go about designing something like this? Well, I think I said, yeah, so we sort of have one set of treatments that we're more interested in than others should tell us that what we want to do is to have a differential level of replication. So we need more information about the treatments that are more important to us, so the 0 and the full, and maybe less information about the minus treatments. We obviously wanted to look at all of the soils. We're interested in comparing all of the soils with each other. That's part of the assessment that was needed. But we can really start looking at some answers, if you like, to how these constraints might be used. So we can identify fairly quickly that the 30 soils need at least 3 trays to actually get the full set of replicates for a single replicate. So 10 pots per tray would be a division of the soils between trays. We've obviously got a little extra space in those pots, so maybe we could add in an additional pot of a standard soil. And the advantage of doing that is that it improves the comparisons that we might be able to make between those, between the soils that go in different trays, in different benches, in different compartments. Another constraint caused by the way the treatment was going to be applied was that the pots would have to be, the pots within each tray would have to have the same nutrient treatment. Otherwise, we have issues of contamination between those treatments potentially in some way. So the approach to designing this problem was really to break it down into 2 parts. A first part to decide how we allocate the soils to the trays, and then a second part to decide how we allocate the trays to the glasshouse compartments and the benches that we have. So the soil allocation, what we used was the idea of an incomplete block design, a so-called alpha design, developed based on work by Desmond Patterson, who was here in the 50s and 60s. And the construction was to do a separate design for each of our nutrient treatments, taking account of the replication levels. And the power of the alpha design is that it arranges the soils so that all pairs of soils occur as evenly as possible across the trays. So we get as equal information as possible for all comparisons of pairs of soils. And so here's just an example of part of that design. So this is the design for one of our 0 or full nutrients, showing the 30 soils, 30 numbered soils, and how they appear in pots across each of the blocks within each of the 5 replicates. You can see the control treatment labelled by 0, which occurs in every tray. As I said, that provides extra comparison between the trays. And I've also tried to illustrate how within the comparisons that we get there, different pairs occur together slightly different numbers of times. So we can see that treatments 1, 4, and 5, soils 1, 4, and 5 occur together in a block in the first replicate, and never occur all together in any of the other replicates, but do occur in pairs in some cases. So 1 and 4 appear together, 1 and 5 appear together, and I think that's it. And 4 and 5 never appear together again. So a sort of a distribution of the soils across the trays. The second part of the design was when working out how to allocate the treatments, the nutrient treatments, to the trays and then to within the benches and the compartments.
Again, this has an incomplete block design nature to it, so we can't get all of our treatments onto one bench because we've got 7 treatments. We have more replicates of 0 and full than of the other treatments. And so we need to find a way of actually achieving this. And the design is not a perfect design. A perfect design would require us to have 5 compartments, and we only have 3. Just the number of replicates of each of the treatments that we've got. Each tray has to have both a 0 and a full treatment in it, and then various combinations of the others. And you might be able to see the pattern, so that's just illustrating that the 0s occur in each of our benches. And that within the benches that only have a single 0 and a single full treatment, we have a common, one treatment that is common, so K in the top block, S in the next block, and P in the third block, that occur with each of the other 4 nutrient treatments across those 2 benches that they occur in. The approach, if you like, what I'm trying to illustrate, I guess, with the approach to achieving this is, I've not used any complicated mathematics to try and come up with this design. If you like, this design is a series of working out what the constraints are and coming up with an intuition about how we put the treatments together to get the best information from that arrangement. The analysis will be quite complicated. We haven't done it yet. It will need us to use this more complicated algorithm, REML, rather than the analysis of variance algorithm. But we can, in advance, do an assessment of how good this design will be for comparing the different treatments that we're interested in. Okay, I'm going to move on to some other examples. So another example of the challenge of designing an experiment is illustrated by this, which is part of a European collaboration at the Coffee project. This is a project to look at genotype by environment by management interactions with a series of trials across 12 European sites. Each of those sites provided a variety, a cultivar for assessment on each of the sites, and we're then also looking at a nitrogen effect. So this is on first sight, seemed like a very simple analysis, simple design to construct. We needed to apply nitrogen to rates to large areas of plots. So we end up with a split plot design with cultivar randomized within those. So it looks like it's very, very simple, straightforward design until the farm get involved. And the farm here at Rothamsted said, well, we can't do that. We've got to, we want to apply nitrogen to strips across the field so that you can see in the illustration on the right-hand side there, the colours show the different nitrogen treatments. But we may also want to allow for variation due to the field management operations. So, for example, drilling is done in vertical strips from top to bottom. Okay, so how do we allow for that kind of information? Well, we still want to have 3 replicate blocks, and hopefully you can see that we have three replicate blocks for the nitrogen treatments from top to bottom. But we also want to have some replication associated with the farming operations. So what we have in this design, in addition, is that each of 3 vertical strips is a complete replicate of our treatments. So each set of 4 columns contains a complete replicate. The design is actually constructed using what's called a semi-Latin square, which you may have come across Latin squares. Latin squares allow for variation in 2 different directions. The design uses a so-called semi-Latin square with a randomly selected pair of cultivars that will always appear together within that design.
So just to illustrate that last point is that if I look at this one particular nitrogen rate, we find that in each of the in the 3 by 3 arrangement, if you like, that we've got there of the two replication structures, that those pair of treatments always occur together, though you probably can't read enough of it to see that they occur with different sets of other treatments. So that these two treatments, Hondia and Julius, occur with 6 of the other varieties within those different, the segments that they appear. So we get information, we get differences between treatments independently of differences between the rows and of the columns of our design. But it gives us a way of coping with the constraints imposed by the nitrogen application and by the other field management operations. So 2 relatively straightforward designs, 2 relatively straightforward problems, or so we might think, but where we need to do a little bit of extra thinking about how to actually use the resources effectively, efficiently, to answer the questions we're interested in. A third example is illustrating the idea of what do we do when we've got more treatments than the resources that we've got available to do the experiment with. So this is an example from Northwick of using Dennis to look at greenhouse gas emissions from soils. Initial discussions, again, thinking about the design was, well, they wanted to look at 6 factors. Eventually decided, looking at the number of factors and the number of levels of those factors they were interested in, that there was far too many treatment combinations to look at, and initial discussions resulted in one of those factors being dropped. But we still have 96 treatment combinations that we want to actually look at. 96 treatment combinations we need to look at to answer the questions we're interested in. The system that we're using, Dennis, has 12 independent chambers, which have to be incubated at the same temperature. And notice that one of our treatments is temperature. And resources in this project only allowed us to actually have 4 runs of the system. So we can only make 48 observations, and we've got 96 treatments that we're interested in comparing, which sounds like an insoluble problem. How do we look at 96 treatment combinations with 48 observations? The answer is we can't. But what we can do is to find a subset of the treatments that allows us to get most of the information about the treatment effects that we're interested in. And this is a very common form of design used in industrial statistics, but seems to be relatively uncommon to date, used within the agricultural world. What we do is we choose combinations of the treatments that will maximize the information we can obtain on the most important model terms. And usually that means the main effects of those factors, 5 factors we're interested in, and the 2-factor interactions. And there are 10 2-factor interactions that we might want to look at. What we end up with is what's described as confounded fractional factorial design. The design is such that temperature is confounded, or if you like, confused between the runs. So if you look in my illustration at the bottom here, and I don't expect you to be able to read everything, that 2 of those runs are at high temperature and 2 of those runs are at low temperatures. So the effect of temperature is confused with run, which means that potentially differences between runs might actually be, differences between temperatures could be partially due to differences between the runs and other things that have changed between those runs. We have to confound some other elements, also with runs. And what we do is we confuse or we confound high-order interactions with those runs to enable us to still get information about the main effects and the low-order interactions. Okay, so the key point of the analysis of this kind of an experiment is we have to make some additional assumptions. We have to make some assumptions that the high order interactions, so the 3 and 4 and 5 factor interactions between our factors, aren't very important. Of course, if that assumption is wrong, then we may be getting out some very confused answers out. But the principle is that we try and we make that assumption and we use that, those high order interactions as our estimate of the error of the variability, which we then compare our treatment effects to. Those who are looking, so on the right-hand side, I've showed the analysis of variance table that we'll get out for this particular design. I meant to point out also, if you can see them, that within each of these blocks, still not working. No. Each nitrogen level, for example, will occur. There are 4 nitrogen levels, so each of the one nitrogen level occurs 3 times in each of our runs. The water-filled pore space, each of the 3 levels of that, so each one occurs 4 times during in each of those runs. There are 2 soil types, each one occurs 6 times, and 2 volumes, each one occurs 6 times. So that within each of those blocks, we can get good information out about each of those main effects because they're equally replicated within each of those runs. The interaction, that we can get the interaction out, is probably less obvious because it's more difficult to see the combinations that occur in each block. But the analysis does allow us, as you can see on the right-hand side there, to get information out about all of the main effects and all of the 2-factor interactions. Although those who are very quick will notice that some of the 2-factor interactions don't appear to have quite as many degrees of freedom as we'd want them to have. Those involving urine N, which has 4 levels, so 3 degrees of freedom. The interactions should also have, in most cases, 3 degrees of freedom, and we lose some information into the block stratum for some of those effects. So hopefully, having convinced you that having less than 1 replicate is actually possible, you're no longer going to be worried about my design for the new long-term experiment, which has 1 replicate. So those of you who are not fully aware of this experiment, so it's a new experiment that has now been established just over a year. And the aim of it was to quantify effects of various combinations of management drivers on metrics of sustainability. After a lot of discussion, this was an experiment designed by committee. We came up with a series of treatment factors, rotation length, intensity and cultivation being the most important factors, but we're also interested in looking at the effects of nutrition and the effects of crop protection. About 5 months ago, I talked at the LTE conference about the principles for designing LTEs, and I've sort of repeated some of those here, that they need to be representative and relevant, so can they be related to farmer practice? The design needs to be flexible, and in this case that meant having large plots, or relatively large plots, having the potential to change treatment levels, having the potential to add or remove treatments at a later stage. And as with any experiment, the design for any experiment needs to be obviously influenced by the purpose of that experiment. A key idea that we identified in trying to design this rotational experiment was that we wanted to have all rotational phases observed simultaneously. And the advantage of that is that we can both look at the effect of treatments over time by aggregating responses in multiple years, and over space by aggregating responses across the plots that are in different phases, but the rest of the treatment being the same. So this, the eventual design that the experiment we were trying to design had 120 treatment combinations, including the different rotational phases, and there were going to be limitations on the resources that we were going to be able to use. As we developed this design, developed the experiment, the primary interest, our primary concern for the experiment was that we were interested in comparing cropping systems rather than comparing the effects of individual factors. So whilst we might think about a factorial system, a factorial treatment structure always makes sense because of the extra information you can get from thinking about a factorial treatment structure. In this case, the factorial treatment structure was designed to generate a wide diversity of systems that we can then compare. And this led us to what I describe as a model-based analysis approach. That is, that we're not actually interested in doing the analysis of variance on the factors that we build into our design, but on relating outputs, possibly multiple different outputs, to multiple inputs, different inputs, if you like, realized from the combinations of the treatment factors that we've used to construct the design. And so this is my main argument as to why we don't actually need more than one replicate of any of the treatments. Because actually, as the treatments develop, as the systems developed, they're not actually going to be particularly closely related to the factors that we've used to define them. So there'll be an adaptation of nutritional treatments depending on what the other treatment factors are actually, how they're influencing the overall system. And we also, it's a long-term experiment, or hopefully it's a long-term experiment, the replication we also obtain by the fact that each year provides us a separate realization of the treatment structure. I'm running out of time. I've probably run out of time. This plan on the right-hand side illustrates the way we started thinking about this design, which was that we wanted to group together plots for each rotation. So we had, in this initial thinking, we only had rotations of length 5 years and 7 years. And that would have allowed us to consider farmlet-level treatments, that is, treatments such as crop protection that we might think of wanting to apply to a whole series of plots that were essentially part of the same system. But various practical constraints, again, meant that, so for example, that different plots, different crops in the sequence would be cultivated at different times, meant that that wasn't a practical approach. And so we ended up with a design that has 60 main plots for combinations of the rotation phase, the cultivation and crop protection, each then split for 2 different nutrient treatments. We could try and analyze it as a factorial structure, and you can try to illustrate here that actually we can get lots of information about the main effects and 2-factor interactions by doing so. We can even build in the effect of the phase of the rotation, although I'm not quite sure how we interpret that. But the principal approach to the analysis of the data from this experiment will be this idea of a model-based analysis relating outputs to a whole series of different inputs. That's the final design, the illustration of the design as implemented at Broom's Barn that has now gone through 1 complete season. This is a picture that was taken in early spring this year, just trying to illustrate the implementation of that design. How much longer have I got, Kim? Okay. So, just again to illustrate that some of these ideas of not having enough resources or not having complete replicates can be applied in other examples as well. So this is another trial that we developed at Northwick. It's part of the ASSIST program looking at different grazing and livestock systems. So using the Rowden plots, we started with the interesting challenge of having only 12 plots. That was all that's available for running this experiment. We wanted to look at 3 sward types in combination with 2 different grazing strategies. Now, that might sound reasonably straightforward. That sounds like only 6 treatment combinations, so that sounds maybe we can have 2 replicates. Unfortunately, there's a constraint, or a number of constraints. We had 6 plots that were drained and 6 plots that were undrained. They're shown on the plan on the right-hand side. The shaded ones are drained and the unshaded ones are undrained. We might also expect there's a path going up the middle of the plots. So maybe there'll be differences between the 2 sides of the path. And another problem when we started thinking about implementing it was that we had three plots that had a very poor pasture to start with, and so therefore needed to be reseeded. So they couldn't get the permanent pasture treatment of basically leaving the pasture as it was currently there. So we had those obvious constraints. We had concern about spatial variation within this trial space, so that we wanted to distribute the sward types and the grazing strategy treatments evenly across the site. And an issue that I have met before, that we wanted to be able to have the three different sward types so that it was easy for somebody to come and look at them and see the differences between them without having to walk too far into the experiment. So down the set of 4 plots on the left-hand side there. So a lot of constraints. How do we solve this? Well, we have 12 treatment combinations to be allocated to 12 plots. So now we're down to a single replicate, in effect, if we take account of drainage. Again, we can make some assumptions about certain of the interactions not being particularly important, or that we consider them to be negligible. So 2 interactions, the interaction between drainage and grazing intensity that we could decide to be negligible because drainage happens in winter, grazing happens in summer. But we still want to assess the other 2-factor interactions. When you look at the maths of this, we realize that there's about 500,000 possible combinations that we might want to think about. So how do we decide which ones of those is the best one to use? And those are the constraints. So the approach here is to use a kind of constrained randomization approach. That is, that we look at generating a series of separate randomizations, in this case 10,000, and identify all of those that meet the constraints that we have, meet the sets of constraints that we've got there. And select at random one of those, in this case 700 randomizations that met all of our constraints. The analysis for a single year is shown in the dummy analysis of variance table there, which shows that if we're only interested in 3 main effects and 2 2-factor interactions, we still have 3 degrees of freedom for our residuals, so we can still estimate the background variation. But the real power of this experiment comes from the fact that we can then look at replication over time, and we can then get interactions with weather effects as well. I think I'm going to skip over the next couple of things. So a similar idea was applied to the allocation of cattle to the 3 farmlets at Northwick, part of the farm platform. This gets a bit more complicated than the previous one, and there are lots of different combinations. It's an application of what's called covariant-based constrained randomization, which is a very common approach used in medical research for looking at design of clinical trials, and essentially is about identifying the constraints, looking at the permutations of possible permutations of our treatments, or allocation of treatments, in this case cattle to the different farmlets, allowing for the best balance of the constraints that we have. So allowing for the different genders of animals, the different breeds of cattle, and a whole range of variability of other variables as well. Again, there's far too many permutations that you could possibly look at all of them. I think it was something like, did I put it on here? No. 10 to the power 50 or so possible permutations to look at, which probably there isn't enough time this side of the next millennium to look at all of them. So again, this idea of looking at a whole series of possible permutations and choosing those that meet the criteria. I'm going to skip past this and just, I think, touch briefly on this last example. One of the challenges, I think, of current research that we do is that often we have experiments that might be thought of as having multiple phases. So a common example of that, but it's certainly not the only one, is of experiments within the omics world. This is an example of an experiment that was done here a few years ago now. I think it was completed before I actually got to Rothamsted. But was about growing trees in various different ways. So we have trees being grown and tipped to try and look at the impact of the tipping on the response. But the measurement of the response is done through an RNA-seq experiment. So there's a sort of a second phase to the experiment. So growing the samples in a field, or in this case a controlled environment experiment, and then processing those samples using RNA-seq. And the idea of these multi-phase experiments, or the challenge of these multi-phase experiments, is how you combine those two pieces of information together. How we can combine what is a relatively simple first part of the experiment, where we have a randomized complete block design for 15 treatments in 3 replicates, combinations of time and other treatments, as you can see in the picture there, and then put those samples into a second constrained system where we can only look at 10 samples per chip. The ideas for some of the ideas behind these designs are developed by Chris Bryan and Rosemary Bailey. Rosemary Bailey was here in the 1980s. And what I've tried to illustrate here, the principle of multi-phase designs, is that we confuse blocking structures between different parts of the design to try and get the best information. So again, we have, in this case, 30 samples per replicate. So that means we have 3 chips to get all of those samples on. And what we want to do is to arrange those samples so the differences between chips don't influence the differences between the treatments that we might want to measure. And on the right-hand side here, what I've tried to do is illustrate that. So the numbers show identify the 3 different chips for a particular replicate. And hopefully you can see that the treatments are arranged so that each time point, the columns, is spread evenly across the 3 chips. And each of the 6 treatment combinations that we've got for the rest of the part of the treatment is also spread evenly across the chips, or as evenly as we can achieve, and therefore gives us the best comparison of those treatments, taking account of both parts of the design. Obviously, depending on the blocking structure, we may be able to use analysis of variance, or we may be able to use a more complicated Remel approach. Well, I just wanted, on this particular example, just to illustrate or talk about the idea of the analysis of omics experiments with a complicated design structure. So the analysis here, the RNA-seq produces count data, which requires us to think about analysis using a GLM. And I think this is a challenge that we're still trying to address, of a conflict between the bioinformatic analysis and the statistical analysis of this kind of data. So a bioinformatic analysis where we're interested in high throughput, where we're wanting to identify significant genes in some way. And a statistical analysis, which tends to be focused more on the ideas of model fitting and identification of the significant factors influencing each of those genes. The complication with this experiment is that it ended up with 9 possible treatment terms that we'd be interested in, which means there are, if you want to think about those as defining the groups of genes that are interesting, that there's around 500 possible different models that we might have for our differences between genes. And we also may need to adjust for multiple testing, so false discovery rate adjustment. And this just, what I wanted to pick up on here is an approach that we've been working on, that Kirsty and I have been working on for the last couple of years, which is looking at ways of combining model selection and multiple testing adjustments to allow us to identify genes with similar predictive models. I think I'd better draw to a close. But if anyone's interested in that particular work, we'll be very happy to talk more about that.
I'll skip past this last one of just illustrating that we can use similar design ideas to design computer experiments to look at sensitivity analysis of computer models, and leave you with some final thoughts. That the development of statistical analysis tools and the increasing computational power that we now have allow us to analyze very large data sets from complex experiments. Okay? But still, designing those experiments to be as efficient as possible and as effective as possible is still really important to pursuing high-quality scientific research. What I've tried to illustrate, I hope I've tried to manage to illustrate, is that there are various different design ideas that we can use, often borrowed from other application areas, that we can use to try and design more effective and efficient biological experiments. But I'll leave you with a quote from Fisher. I started with Fisher, so I'll end with Fisher, about consulting your statistician. This quote from 1938 was that to consult the statistician after an experiment is finished is often merely to ask him to conduct a post-mortem examination. He can perhaps say what the experiment died of. I like to think that the modern tools that we have maybe means we can resurrect the odd experiment. But it's still better to get the design right in the first place, because that makes resurrection unnecessary. So I'd encourage you to think about involving the statistics group throughout your research programs, from the initial planning, the design, all the way through to the analysis, modeling, interpretation, and the presentation. And I'll just leave you with the acknowledgements of the various people with whom I have interacted on those various examples. And I should emphasize that the interpretation that I've put on all of this is my interpretation. And if I've misinterpreted or misrepresented any of the people I've worked with here, it's my fault, not theirs. Thank you.